Good afternoon, grade 12s. Welcome to our revision session on Term 2 work. Today, I will be helping you guys through the questions on genetics. I am Yolanda Gibson, a teacher at Parklands College, and I hope I will be able to get some clarity on some of the areas that you might be struggling with. So let's dig into a revision session on the genetics concepts, and we're starting off with the types of dominance. So you guys would have seen in one of our earlier videos from this year that we discussed the types of dominance in detail. So our first one that we have here is complete dominance. So this is a case where we have, for instance, a red flower which crosses with a red flower, and we can see that there will be red flowered offspring. And when we look at the description of what is complete dominance all about, we see that it's a case where one allele is dominant and the other allele is recessive. So remember that an allele is indicated by one letter. So in this setup, we've got two alleles. The capital letter will be the dominant allele, and the small letter will be the recessive allele. So when we read the definition further, it says that such that the effect of the recessive allele is masked by the dominant allele in the heterozygous condition. So in this case, we've got the capital letter that's dominant over the small letter, recessive, and we find that this red flower does have genetic information for the recessive characteristic, which could be a white flower, but because of that one allele being dominant, it will only result in the dominant phenotype being expressed. So when we have a case where these two flowers cross and we look at one of the offspring, we see that there's complete dominance at play. A red flower and a red flower will end up giving us a heterozygous offspring where that red phenotype is present yet again. Right, with incomplete dominance, we have a different scenario. So where complete dominance could have been a red flower and a red flower giving a red offspring, or a white flower and a white flower giving white offspring, or we could have even a red and a red giving us red and white offspring. We see an incomplete dominance, a case where our flowers, when they are crossed with each other, a red and a white one, will give us a pink color. So I like to think of this as that it's incomplete, it's neither red nor is it white, it's an intermediate color, so incomplete, intermediate color, which is represented. And if I take my paint and I take a little bit of red paint and a little bit of white paint and I mix it together, I'm going to get that intermediate color, which is like a lightish pink. So when we look at the description, it says none of the two alleles of a gene is dominant over the other, resulting in an intermediate phenotype in the heterozygous condition. So the letter R and the letter W, neither of them is considered to be dominant or recessive. They are both, in this case, trying to express their own phenotype here, and they're not achieving it. So we end up getting an intermediate color, which is then pink. For our last one, we have codominance. And for codominance, I think of the word co, and immediately what comes to mind is the word co-pilot. And that's my association that I make. When we have two co-pilots flying a plane, they are both equally in charge of getting the plane to where it's supposed to be. So when we look at the flower cross, we can see that when we have a red flower and a white flower that crosses, in the offspring, we will find that there's a blotchy red and white flower present. So that means that these two alleles that we have here, they are co-pilots in expressing that phenotype, where both of them have achieved to express themselves equally in the phenotype. So when we go to the definition of it, it says both alleles of a gene are equally dominant, whereby both alleles express themselves in the phenotype of the heterozygous condition. And remember, heterozygous, as we've seen in the previous example, is where we have two letters representing two different letters. So here we've got an R and a W. This will be considered homozygous. Homo means the same. Our second revision session is about Mendel's principles, so I'm going to go through them quite quickly. We're going to start off with Mendel's principle of segregation, which states that the pair of alleles on homologous chromosomes separate during meiosis gamete formation so that only one allele of each pair is present in the gamete. So when we have two chromosomes next to each other, those two chromosomes will separate from each other through random arrangement, and then we will have the two alleles on opposite gametes. Right, the second part, Mendel's principle of independent assortment. It states alleles of a gene for one characteristic segregates independently of the allele of a gene for another characteristic. The alleles for each gene will therefore come together randomly during gamete formation. So that will then be our independent assortment. Our last law or principle states Mendel's law of dominance, which is when two organisms with pure breeding, contrasting traits. So when something is pure breeding, it means, like in the case of flower crossing, we have a red flower and it crosses with the red flower. All of the offspring will then be red, and every single time that you cross it, that means it's pure breeding. And the same applies to a white flower. If I have two white flowers and I cross them and all of the offspring are white, it's considered a pure breeding flower. So if we read this again, it says when two organisms of pure breeding, contrasting traits, so a red flower and a white flower is crossed, all of the individuals of the F1 generation, so the first offspring lot that we find, will display the dominant trait. So for instance, if we have a red flower and it crosses with a white flower, and we see that all of the offspring are red, then we know, okay, in that scenario, that red was the dominant phenotype. So that is all about Mendel's law of dominance, or Mendel's principle as it's also known. Right, so let's dig into what we can expect in an exam paper. Now, you will be asked to set up monohybrid genetic problems. Monohybrid means one characteristic that we are going to look at a time, and we want to know how the offspring would look from a particular cross. So you would need to learn this off by heart. This will be the backbone that you would use when answering a typical genetic diagram question. So the P will then refer to the parents, the parental phenotype. I tend to write it again, P1 genotype. Genotype will refer to the alleles. Then it's like a story. So when we have our parents and they produce children, it first needs to undergo, well, in order to get a child, you first need to undergo meiosis to form gametes. And the gametes will fuse together through the process of fertilization to form the offspring at the end. So that F1 refers to the filials, the offspring, which we will then look at the F1 genotype, allele combination, and then the F1 phenotype, which is the physical expression of the allele combination that we have. On the right-hand side, for those of you that do not like the line crossings, you can set up a little Punnett square as part of your answer. So we will be looking at the two examples during the session, and you can choose which of these two methods you find to be more suited to your learning style. Right. Now, our first example that we have is in dogs, rough hair is dominant to smooth hair. So we have two examples here. So we've got Jack Russell's. The Jack Russell on the left-hand side's got rough hair. The one on the right-hand side has smooth hair. So the question says that a heterozygous roughed hair mated with a smooth hair dog. So this individual's genotype will be that it's heterozygous. So that means hetero is different. We will have a capital H and a small h. And this will be the expression, the phenotype of such a gene combination, allele combination. Now, on the right-hand side, we've got our smooth hair individual. And because this is not the recessive characteristic linked to a small letter, that's how we know it's recessive, because recessive is always indicated by a small letter. This dog's genotype will then be small h, small h. So they want us to represent a genetic cross to show the phenotypic ratio of the puppies. So we're going to start off by writing out our backbone. This is now for a line diagram, if we're going to make use of the lines instead of a Punnett diagram. So we're going to start by writing out what the parentals look like, the P1 phenotype. So that's directly from the story. We had a rough-haired dog crossed with a smooth-haired dog. So we fill that in. Then we want to know what the genotypes would be. The story tells us it's heterozygous for the one, and the other one is a smooth-haired dog, so it has to be recessive alleles. So homozygous recessive on the right-hand side here. Then the story needs to continue. You have to write. We want meiosis to occur in order to form gametes. So that will be an empty space that we leave there. And then we write down what the potential gametes for each of those two dogs would be. So for instance, in a male, we will find that there could be a capital H and a small h. And then for the smooth-haired dog, the gametes that form can only be two small h's. So this guy's been a little bit too quick for us, but when you have your gametes, the next step will be to indicate fertilization. So fertilization will be written down where the lines will be found in the diagram as we see here. Then you're going to fill in next to the F1 genotype the first consequence of a gamete coming from the male dog fusing with the female gametes to form a zygote. So then the allele combination will be indicated here as capital H, small h. When we have our second gamete from the rough-haired individual fusing with the second gamete of the smooth-haired individual, we end up with a homozygous recessive offspring, represented in this diagram already as small h, small h. We draw our third line, and what we find is that the first gamete, when it fuses with the first gamete of the smooth-haired dog, we will find a heterozygous offspring. And for our last cross, when we have our second gamete of the rough-haired individual combining with the second gamete of the smooth-haired dog, we will find yet again another homozygous recessive puppy. So when we analyze what we've got in front of us, we will see that there will be two representing the dominant phenotype and two representing the recessive phenotype. So when we have to fill in the phenotypic ratio or phenotypical ratio, we are not writing down 2 to 2, we are reducing it down to 1 to 1. So always remember that you're not going to leave it as 2 to 2, reduce it, and then write next to it 1 rough-haired puppy to 1 smooth-haired puppy. And indicative of a ratio, you have to have that sign in the middle between the two. And then basically the mark allocation for this typical question would be P1 and F1 receiving a Mahala mark there, and then for meiosis and fertilization being written down, you get the second one. And the other 4 marks will be distributed either as the P1 genotype or P1 phenotype at the top, or the gametes, or for having the correct final answer. Our second question, based on genetic diagrams, is based on a fish example where we have 3 phenotypes representing fin length, an elongated, a short, and a medium fin length. Now, the story tells us that the heterozygous fish have medium fins. This characteristic is under control of 1 gene with 2 alleles. So here we have an elongated E being represented by the letter E and a short fin represented by the letter S. So the question wants to know, name and describe the type of dominance that is shown here. So when we have 2 letters, and these 2 letters are obviously now a big letter like an E and a different letter, an S, we are immediately working with either incomplete dominance or codominance. So then you have to look, okay, which of the 2s are going to be? Are we working with a case where we have an intermediate phenotype, or do we have both being expressed in the scenario? So that's why you have to know the different types of dominances really well. And in our story on the side, it told us that we have medium fins, we have elongated, and we've got short fins. So the medium fin is then an indication that we have the intermediate phenotype. So that's linked to incomplete dominance. So when answering the question, when we have our memo in front of us, we have started with incomplete dominance being the answer. We've identified that. But the question's asking us to also describe the type of dominance which is shown here. And therefore, we go to the second part. It is a genetic cross between parents of elongated and short fins, and then basically two different phenotype parents that will then produce offspring of medium fins, the phenotypical difference of both the parents, resulting in that intermediate phenotype that we were talking about. So that's question 4.1. Next, I want to know, use the genetic cross to show the percentage chance of two fish with medium fins having offspring with short fins. And in this example, we start off by writing down the phenotype from the story, and lucky you will get a mark just for writing that down next to P1 phenotype. Next, they want to know the genotype of this cross. And because we are working with medium fins, all right, both of them will have the intermediate phenotype here. It means that we have to have a letter E and S present in each of those two genotypes. Now, we are showing a separate, different cross here. This is no longer the line cross that we were practicing in the previous question. We are now including a little Punnett diagram here on the side. So take note that we are still sticking to our backbone here on the side. So we're going to have P1 phenotype, P1 genotype. I would shift that meiosis a little bit up so that it's above your Punnett diagram. Then you have your Punnett diagram with the word gametes included in it. And from there, we will have fertilization being represented next to it. And we've got our F1 phenotype at the bottom. So when we look at this question and the gametes are now combining with each other, we will find that the two, remember that the top line represents one parent and the side represents another parent. So when we see the two letters EE next to each other, that's one of the potential offspring that could be formed. Now we do it for the next offspring that could be formed, potential one, and we see it's ES. The third potential offspring, ES yet again, and the fourth potential offspring that could form is SS. So what a Punnett diagram is showing us is that we are looking at 4 possibilities of genotype combinations that could occur. So we're not saying that there will be 4 offspring and each of those 4 offspring would have to have one of those 4 genotypes. We could have a case where all 4 of the offspring have the exact same genotype because it's all about which sperm cell got to the egg cell first. So let's analyze a Punnett diagram to see the final offspring combination. Now, when we have 2 E's next to each other, we have elongated fins. When we have ES next to each other, it's incomplete dominance. So we have the intermediate phenotype that will be medium fins. Here's another medium finned offspring. And there we have a short finned offspring. So when we look at the phenotype, there's a 25% chance of having short fins because they were only interested in knowing how many medium fins will be present. And we know that when we look at this, that that in this case we're looking at short fins, actually not medium fins, that will only be that 25% which is represented there. Okay, next up we have blood groups. And for this particular question, a man which is heterozygous for blood group A marries a woman which has the blood group O. Each use a genetic diagram to show the phenotypical ratio. So we start off yet again by writing our phenotypes here. But it's important that you guys remember that when we are working with blood groups, there are 4 different types of blood groups. So we have blood group, we've got A, we've got B, we've got O, and we have AB. So the genotypes that can be linked to your A blood group is IA, or it can be capital I, A, and a small letter. Or for B, we can have IB, or it can be B, and a small letter O. For the O blood group, there's only one genotype that's linked to it, and then for AB blood group, there's also only one genotype that is linked to it. So this you would need to know off by heart before you're able to answer this particular question. So if you don't know what the potential genotypes are for your A blood group or your O blood group, you will have a tough time answering this question. So in the story, they have given us an example where we've got an A blood group father and an O blood group mother. And they want to know what are the possibilities of the offspring being formed. But they also tell us that we have a heterozygous A blood group father. So that means that we are working with a capital I, A, and then a O allele right next to it, which is represented by a small I. So if we fill in the blood group for a father, we can see that one allele being representative of the A blood group, and then the O allele right next to it. But here we've got complete dominance, so that allele, A allele, will then actually express in the phenotype. For our O alleles, it's represented by two small i's. When we do the cross, yet again, I would have gone and put that meiosis slightly more upwards into the space that we have here so that it follows the story. And then gametes, remember to fill that in, and then we have fertilization here on the side. So when we go and we complete this diagram, the Panna diagram, with the one parent in the top row and the other parent on the side, and we fill in the combination of the potential offspring, we will see that the F1 phenotype will result in two blood groups being of the A type and two blood groups representing the O blood group type. And because we reduce our answer, the phenotypic ratio will be 1:1. Our next example that we have is hemophilia, which is a sex-linked disease, which I'm sure that you have been exposed to in classroom about hemophilia as a blood clotting disorder. And this is usually always found on your X chromosome. Now, it's specifically linked to your X chromosome and not the Y chromosome, and the reason being is that the Y chromosome is much shorter than that of the X chromosome. The consequence of that shorter chromosome is that there's genetic information which is missing. And in the case, this will be that there's not a healthy allele that could mask the effect of a disease allele if it's inherited on the X chromosome. So let's have a look at a question. I'm getting so excited here. Let's have a look at the question. It says a hemophiliac female marries a normal male. Explain why all of their sons will be hemophiliacs. So when we go through the answer for this question, we need to start off by saying an individual inherits one allele from each parent, because remember there are 46 chromosomes. One chromosome set will come from each parent. And in this case, we are interested in a son and why the son will be a hemophiliac. So he will receive his Y chromosome from his father, and he will receive the X chromosome from the mother. But now, if the child receives a diseased allele from the mother, it's represented always by an X and then a small h attached to it. That shows that it's a recessive disease. Then our next part says that since the mother has two recessive alleles, a son only needs to get one recessive allele to be a hemophiliac. And the reason being is that the Y chromosome does not carry the allele to mask the effect of the hemophiliac allele. So as we've already established, when it comes to sex determination in children, your father will always pass down a Y chromosome to the son, and the mother will then pass down the X chromosome to the son. So in that case, if the mother is a hemophiliac, there's no other way around it. She will be passing down a recessive allele to the offspring, and that poor child will then express the mother's disease allele because the Y chromosome will not have a healthy allele attached to it. Right, so let's apply this question quickly. So in a sex-linked disorder, in this one, question 7, they are linking it to rickets. Now, your teacher has not forgotten to teach you about rickets. It can be a case where you are given some random disease that you've never heard of before. Don't get a fright. Your teacher's not neglected you in your teaching. We ignore whatever disease they've then mentioned if it's not familiar, and we read the rest of the story, and it will tell you something like, and then it is a sex-linked disease. So we know to use the letter X and Y to represent it. Now let's go into the question. And for this question, a bit too soon, in this question it says, determine the possible genotypes and phenotypes of their offspring by representing it as a genetic cross. So what I'm going to do is I'm also going to read the next two questions because we're going to answer it as a whole. 7.2, what is the percentage chance that they will have a child who is an unaffected male? And for 7.3, explain why the disorder, although it's sex-linked, does not affect males only. So looking at our answer to this question, because it is now linked to a dominant allele in this case, they've given it as the scenario, which is linked to the X chromosome. That's very kind of them. They tell us that it has to be a capital R linked to an X. An affected female whose father was unaffected is marrying an unaffected male. So if it's an unaffected male, we fill that in, and an affected female. And because they have told us that the female comes from a family where the father was unaffected, it's telling us basically that the father has given this female an X with a small r attached to it, because unaffected is represented by X small r. So the dad definitely gave that to the offspring, and this mother inherited that disease allele from her mother. Okay, so the next part will be what is the unaffected male's genotype, and we find that it's X small r Y. So when we look at the Punnett diagram, when we fill it in, and we look at the offspring that can be formed and we analyze it, we see here that that's going to be an affected female. Here we've got an affected male. Here we've got an unaffected female and an unaffected male. So when we write out our phenotype, F1 phenotype, we're going to write exactly that down. So you get your mark. And because they wanted to know in the previous question, the percentage, that will give us 25%. And for 7.3, it is caused by a dominant allele, because the story actually told us that it was a dominant allele, which is carried on X chromosome because it's sex-linked with both males and females, which both males and females would have. And for female to have this disease, she would just need to inherit 2 alleles instead of 1. And question 9. In this one, we are looking at a dihybrid cross. So we are looking at 2 characteristics at the same time. We have red fruit and we have white fruit, with the letters representing the dominant and the recessive characteristic. And then we're also looking at the second characteristic, which is spiny leaves and smooth leaves, with spiny being dominant and smooth being recessive. The Punnett diagram has been given to us, or the Punnett square has been given to us, and we start off with answering our first question, where they want to know what is the genotype of X which has not been filled in. So we have seen that in order for the gametes to combine, we have to use convention, which states that the same characteristic has to be rewritten next to each other, meaning that the R's need to be written next to each other and the L's need to be written next to each other. So when this R and that R combine, we have 2 capital R's. when this L combines with that L, we have two capital L's, which gives us our genotype, capital R, capital R, capital L, capital L. For 9.1b, the phenotype of the parents, and in this case, we would find that the parents are red fruit individuals. We know that the parents are red fruit because the capital R is present in the gametes. And then for the second part, the spiny leaves, because the capital L yet again is present in the offspring. So that parent was heterozygous for spiny leaves, and in this one, the parent was homozygous for red fruit, and this parent homozygous for red fruit, are heterozygous, and then heterozygous for spiny leaves. For 9.2, they want you to apply yourself a bit here, and they are giving us in this hypothetical question that there are 128 offspring plants formed. How many plants will have red fruit and smooth leaves are expected from this Punnett square above? Show all working out. So in order to answer this question, we're first going to look. Red fruit is represented by a capital R. So irrespective of if it's homozygous dominant or heterozygous, we are going to look for any individual that has a capital R present. And then for the second part, smooth leaves, and we know that smooth leaves are present with L. But you have to have two small L's next to each other. So any genotype combination where there's a capital R present and two small L's. So that's where you get your highlighter out and you go on a hunt and you find those individuals that have the heterozygous or homozygous R's and then the recessive L's. So I've circled them for you guys already. So that's 3 out of 16 potential offspring that could form will have this phenotype. So that's a 3 in 16 chance of them having this. So when we apply this ratio or this potential chance to 128 offspring that has been formed, we just do a simple math question where we say, okay, there's a 3 in 16 chance of them having that characteristic. So 3 in 16, if we apply to the 128 offspring that has been formed, that will then be equal to that portion being 24 individuals representing the phenotype of interest. Then 9.3, a farmer wanted to produce plants with only white fruit and spine leaves. Give the phenotypes of the plant that he should use in the cross. So then we find that the answer will be small r, capital L. And basically what we find is that this could be the one answer, plain straightforward, or one parent is small r, capital L, and the other parent can be small r, capital L, small l, because both of these genotypes will give us the phenotype that we are interested in. Okay, we move on to our next question, which could be a genetic diagram, family history, like a phylogenetic tree. And pretty much we have unknowns in this question. So this question is pretty easy in order to work out. They will give you a key with the colored in sections meaning different things. So in this one, we've got one which is not colored in. That's an individual without SCID, which they indicate in this example as being immune deficiency syndrome. So when we have a colored in block, it will indicate in this example as being a male with SCID if it's a square, and if it's a circle, it's a female with SCID. So my starting point will be to fill in the genotypes in the key already, because they will be pretty straightforward in order to establish. In our story, they've already told us that it's caused by a sex-linked recessive allele, and they've given us the example of it. So when we have an individual which is diseased, if it's male, there only needs to be one X small d present, because the Y chromosome doesn't have that allele present, so we just write a Y chromosome next to it. For the female, if she is affected, she needs to have 2 of them present. If the male is unaffected, it will have an X capital D, Y. And if it's a female, sadly for this one, there can be 2 combinations that are present, because this is now complete dominance. And in this case, obviously, if there's a homozygous dominant setup represented by those Ds, then that will express them without. In this case, having that one recessive, one dominant, that one dominant allele is actually masking the effect of the diseased allele. So luckily for that individual, they will be a carrier and they would not express the disease. So what we find is we can actually go to our diagram, and based off the key, we can fill in what we already know. So the green represents everything that I've just taken from the key straight off, that I could just decipher. Okay, now the parts that have not been filled in, I need to look at the information that I've already figured out from the key, and then pretty much work back up in order to answer it. And in this case here, we see that there's a individual that is diseased. We see that the mother was diseased, the father was healthy. So we want to know, for instance, what is that circle's genotype? And what will happen in this case is the father will pass down his dominant allele, X capital D, down to the child, the daughter, and the mother will pass a diseased allele chromosome down to the offspring. So when we fill that in, we will see that the daughter in this setup will have the genotype X capital D, X small d. We can then move back to the top here. And because we have a mother which is unaffected, we already know, okay, well, you know, she's got a diseased allele child on this side, a normal child on the other side. So she has to be heterozygous, because this child received a diseased allele from the mother and a diseased allele from the father. So that would mean that this block that would have been here at the question mark would have been a colored in square to indicate that the father was diseased in order to pass down the second disease allele to that offspring. On this side, because the father is passing down a disease allele to his daughter, the mother had to pass down a healthy allele to that daughter. So the genotype will then be X capital D and X small d for that particular offspring. And then for the rest of it, we will see that it's exactly as I explained it earlier. Now I've taken a screenshot of that to include here on the right hand side. We haven't even answered any of the questions that the question paper has given us. So once you've figured out what's going on in this family tree, we can start to answer the questions. So for question 11.1, they want to know the phenotype of individual 2. So individual 2 is then X capital D small d, and the answer would then be that the person doesn't have a disease. And that's pretty easy, because we could have also just looked at the key, and the key tells us it's a person without SCID. So that's a free mark that we get just for being able to read and interpret the key. For the next one, they want to know what is the phenotype of individual 1. But for this one, we had to go and figure out all of this in order to get to that answer. So X small dy, if we look at our key, that is a male with SCID, so we can fill that in. The genotype for individual 3, question C, if we find question individual 3, we see that it's X capital D, X small d, it's a female, so we're going look, and that is a female without SCID. But they want to know the genotype, which we've already worked out, so we can just fill that in from the diagram. For 11.2, explain how individual 5 inherited this order. And pretty much what we will find is that he inherited the recessive allele from the mother, which is individual 4. And that brings us to the end of that question. Now, a little bit of history that you guys need to go through, or revision from our earlier grades, specifically in history. Grade 10 is a very long time ago. You need to know about cloning. So in cloning, we have a case where you have a characteristic that you have of interest, and you want to have the offspring representing those characteristics. So let's have a look at the process of cloning quickly. So in cloning, what we find is that there will be donor sheep, and in this case here we have the donor sheep giving a nucleus to our egg. And in this example here, we've got the female sheep. She's going to give an egg cell. But we want to clone this guy here at the top. And what will happen is that we are going to extract the nucleus from the ovum, from the donor egg cell, and we give the 2 cells an electrical shock, which will allow for that nucleus to fuse into the empty ovum. And then through the process of mitosis, we will start the zygote and embryos to form. So we will have a ball of cells, which we will then take and we will place it into any surrogate foster mother. And then after a couple of months, we will find that that sheep's going to pop out looking exactly the same as our donor sheep at the top. So this is the history behind our cloning process. Now, when we look at questions like this, they expect you to be able to apply it, so you have to go through that section again. And we have South Africa's example of our first cloning case. So there's a whole story that they discuss. They say the first cloned animal in Africa, a calf named Footy, was born in the northwestern South Africa on the 19th of April. No fertilization was involved in the production of Footy. She was produced from a single cell taken from the ear of a donor cow named LMJC 865, what a name. The donor cow had a high average milk yield of 78 liters a day. So that's why the farm decided to clone that cow, because that's a lot of milk that cow's producing. Now, cloning allows for the production of organisms with the desired characteristic. Obviously, the bonuses is that we're getting that characteristic in all of our offspring then. Some people argue that cloning reduces genetic variation in the offspring. With no further genetic improvement, cloning is an expensive procedure and may not be economical for commercial agriculture. When we look at the questions, it's asking, according to the extract, state one advantage of cloning and one disadvantage of cloning. So for this question, remember that here we are working with a scenario-based situation, and we have been given an extract, and you have to go and find what is the correct answer and rewrite that in your answer. But try and use your own words, perhaps. Do not just go and rewrite the whole paragraph. It's your job to find what is relevant, and they're not asking for the whole section. You need to isolate the important bits. So for the first one, they want to know the advantage of cloning. So let's have a look at the advantages. So first off, we have established that we want that high milk yield. So a cow or an organism that has a desired characteristic. So that's what we're going to get from cloning an individual. We're going to have that desired characteristic being present in the offspring. For disadvantages, we will find several disadvantages linked to it. It reduces genetic variation because all the cows will look exactly the same. There's no further genetic improvement because we have no animals crossing out, making sure that our gene pool's healthy. It can be expensive because we're getting scientists involved here in the lab, and then obviously the vet as well. And then lastly, it might not be economical for commercial agriculture. The question continues. So it's asking, state why the donor cell was taken from our cow with the number and not from any other cow. And that was pretty straightforward because cow LMJC for short, that's the one that had the characteristic that we had of interest, and we wanted that high milk yield production. For 15.3, they want to know step 1, ear cell was used and not an ovum. Remember that a body cell is diploid while an ovum is haploid. So we are skipping the process of fertilization and cloning. So therefore, the answer would be a diploid cell was needed because it already contains all the genetic information which is needed. And also, we have to state why we are not using an ovum. An ovum is haploid and only contains half of the genetic information. Lastly, briefly describe the process of cloning, and when we move through it, we start off with the ovum, which is removed, and it's replaced with a nucleus of a somatic donor cell. Somatic means body cell, which obviously is a diploid cell, and it will form the zygote, which is then stimulated to undergo mitosis, which is the formation of more cells that are identical. And then we will end off with the embryos placed inside the uterus of an adult female and allowed to actually form more offspring. Our last question for today is linked to the process of forming insulin through genetic engineering. And in this question, we find that we have our human cell and we want to form insulin. So what we will do is we will extract the gene associated with insulin production, take it out of a human cell, slice out that segment of DNA. We will take a bacterium, take its genetic information out, make a little entry point for that DNA to be slotted in, and then we would place it back into the bacterium. So that will then end up going into a fermentation tank, which will allow the bacteria to reproduce, and the scientists will go and extract the insulin that is produced by that bacterium. Now, we have fooled that bacterium into thinking that that DNA which has been inserted is its own DNA, but in actual fact, it's human DNA, and it's reading the instructions on how to make a protein, which is directly and exactly linked to human protein synthesis production for human insulin. So when they produce the insulin, it can be extracted from the fermentation tank and bottled, and it will be issued to humans, and it will be an exact replica of human insulin. So they asked us to define the process of genetic engineering. And genetic engineering, when we write it out, it's the manipulation or transfer of genes from one organism to another to make a product that satisfies our own human needs. So in this case, we have taken a gene from a human and we've inserted that into the bacterium. We want the insulin, that's our need that we want from this genetic engineering experiment. And obviously the bacterium is just going to make it for us. So instead of harvesting humans for insulin, we are using the bacteria to make the insulin for us. Question 16.2. Describe the steps involved in producing the recombinant DNA. So recombinant DNA, if we take that word, it's the recombination of DNA. So if we go through the steps, a plasmid is removed from a bacterium cell, cut using an enzyme. So here we can see that there's a little gap there. An enzyme has opened that up for us. And then what we will find is that the human gene will be inserted into that gap. And now we have a plasmid, which is pretty much a recombination of bacterial DNA and human DNA. Question 16.3. Explain why bacteria are the most suitable for genetic engineering. And the reason being is that bacteria can reproduce rapidly, and they produce many copies of the gene in a short period of time. So we can just provide them with all the nutrients that they need, and they just reproduce without any hesitation. Question 16.4 suggests 3 objections that some people might have to genetic engineering. One of them being, yet again, that it's expensive. We might be interfering with, or we are interfering with nature, which might be immoral, that people will find to be immoral. There will be potential health impacts because maybe there's not enough research to prove that it's safe. And we are unsure of the long-term effects because there's not enough research over many years to prove that it is actually safe for human consumption. So these are the potential answers that we can use. So that brings us to the end of our revision pack for term 1. I hope that we have been able to help you guys understand the concepts better, and we wish you all the best of luck for your end of year examinations. We will see you again for our term 2 revision at a later stage. Goodbye.